Good day, dear student. In this pre-lab, I will explain one of the most important biochemical tests, which is Catalase Test. I will discuss when this test is used, the aim, the principle of the test. All this explanation will be presented slide by slide, and I will also show you the video about the procedure of the test. In the lab, we can study biochemical tests that are used for identify and differentiate between bacteria species, because each bacteria produce specific enzymes that help in survive in the environment, and by detecting this enzyme we can differentiate between bacteria species in the lab. For example, two bacteria may look the same under microscope, but one produces the enzyme catalase while other does not. Catalase test can help to differentiate between them. Now, each microorganism prepare a specific substrate and release a specific enzyme. What does this mean? As I mentioned, each bacteria have a specific enzyme helped in survive in the environment. How can differentiate the bacteria according to this enzyme activity? By add the substrate to the media, which is designed to produce a positive reaction. Now, if the bacteria have this enzyme, release this enzyme, they break down the substance in the media, and the reaction can be detected by the color change using the pH indicator or the bubbles formation, for example, at the reagent. If bacteria break down the substance and change the color of the media, this is the positive reaction. So what we mean by biochemical test is a lab method used for identify the microorganism and differentiate between them according to the enzyme activity and chemical reaction. These tests show how the bacteria metabolize or interact with a substance. For example, some bacteria can break down hydrogen peroxide while others cannot. How can differentiate between them? By add the hydrogen peroxide. If they are positive reaction or negative reaction, we can differentiate and identify the bacteria species. Now, one of the most important biochemical tests is the Catalase test, and this test can be done by add the reagent. As I mentioned, biochemical tests we can detect by two methods. By the pH indicator, use a color change in the media, we can detect the reaction, or by the add reagent. In this test, we use the reagent. Add the reagent, hydrogen peroxide, to detect if the bacteria have the catalase enzyme or not. And the aim of this test, we use to differentiate between gram-positive bacteria, gram-positive cocci bacteria, which is staphylococci and streptococci, and gram-positive root-shaped bacteria, Bacillus and Clostridium. In a cocci shape, gram-positive cocci shape, the staphylococci produce the catalase enzyme. It means it's catalase positive, while streptococci is catalase negative because they don't have the catalase enzyme. And we can also use to differentiate between the Bacillus and Clostridium. Bacillus is a catalase positive, it means they have catalase enzyme, while Clostridium is a catalase negative, that means they don't have catalase enzyme. How can we detect if they have catalase enzyme or not? By add the reagent in a colony of the bacteria, which is hydrogen peroxide. So the principle of this test is the catalase enzyme break down hydrogen peroxide into the water and oxygen. When mix the colony of the bacteria with hydrogen peroxide, the procedure of this test we can done by many methods, slide method or tube method. In a slide method, the procedure includes, first we put 1 drop of 3% hydrogen peroxide in a clean slide, 1 drop of the hydrogen peroxide, then use the loop to transfer the bacteria from the fresh culture. Mix the colony of the bacteria with the 1 drop of the 3% hydrogen peroxide. The result, positive test, will be indicated by the active bubbles, while the negative test, there are no bubbles. And bubbles will be appear quickly, which is indicate the positive result. In a tube method, we will add 1 to 2 milliliter of the hydrogen peroxide in a clean test tube. Then add a fresh bacteria colony and observe for the bubble formation. The positive will be active bubbles, negative no bubbles, and strong bubbles in the tube it indicate the catalase positive bacteria. Now, the procedure of the catalase test. As I mentioned, the catalase test used to differentiate between the gram-positive cocci bacteria, staphylococci, and streptococci. Both of them is gram-positive. It appears blue under microscope, and both of them is a round shape. It means cocci. We can differentiate between these two types of the bacteria by catalase test. First, we prepare all the requirement of the test: clean slide, dropper, and loop. We should use plastic loop or stick. We should not use the iron loop, and we need the hydrogen peroxide reagent. The procedure includes a drop of the hydrogen peroxide. I put on a thin slide 3% of the hydrogen peroxide, drop of them. We will add on a clean slide. After that, I take touch of the colony of the bacteria and mix with the hydrogen peroxide. If the bubble appears immediately, it means it's positive reaction. This is the positive reaction because I take the staph colony. Well, if I take strep colony and mix with the reagent, the bubble will not be appear because it's negative. It not have catalase enzyme. That's all about the procedure. It's very simple procedure used to differentiate between staphylococci and streptococci. Now, how can read the result of the test? As I mentioned, the bubbles, it indicate the positive reaction. It means the bacteria have the catalase enzyme such as staphylococci. And no bubbles, it indicate the negative reaction. It means the bacteria, they don't have the catalase enzyme such as streptococci. As I mentioned, the aim of this procedure to differentiate between the gram-positive bacteria, staphylococci, and streptococci. Both of them is gram-positive and cocci under the microscope. How can differentiate between them? By the catalase test. Because one of them have catalase enzyme and other they don't have catalase enzyme. Staphylococci, it have the catalase enzyme, and streptococci, they don't have the catalase enzyme. And the test, it indicates by the bubbles formation immediately after mix the bacterial colony with the hydrogen peroxide. And the limitation of the test, to avoid the false result, we should remember that we not take the colony of the bacteria from the blood agar, because the red blood cell contain the catalase. You don't use blood agar because this may give bubbles even if the bacteria are catalase negative. And we should ensure that the reagent will be fresh, and we don't use the iron loop because the iron might react with the hydrogen peroxide and produce false bubbles. Therefore, always use wooden stick or plastic loop to get the accurate result. And we always use a fresh culture for all tests because the old culture of the bacteria can lead to a false negative result. And in conclusion, this test is a simple, rapid, and very important in bacterial identification, and it commonly used in a medical bacteriological lab to differentiate the gram-positive cocci and other bacteria. It indicated by the bubbles formation when we mix the bacterial colony with the hydrogen peroxide. That's all about this lab. Thank you for your attention, and please, if you have any question, I can explain them to you during the lab.